just generally a more hands-on experience that has mixed wonderfully with the more mathematical aspects of my PhD. So today I'm going to be talking about how we have developed our model for kelp using dynamic energy budget theory, and how our model says that Laminaria hyperborea might respond to this. So we chose to model kelps due to their ecological and economical importance. Once we decided that we were interested in kelp and that they were important, the next obvious stage in a process-based modeling was to identify the key biological aspects of this species. So in kelps, this is their ability to store biological compounds, such as carbohydrates and proteins. This gives them a higher tolerance to environmental fluctuations. In Laminaria hyperborea, this capacity is also used to adapt its fast-growing period. So this species stores carbon in the summer when light conditions are ideal, obviously, and then it uses this to grow in the colder months when nitrate is abundant, but light in Scotland is not, as we are all very aware. So to be able to accurately model those key traits, we chose to work from the dynamic energy budget theory framework. The DEB framework applies differential equations to represent the rates at which energy flows through the individual. It includes the rates at which energy is used for metabolism, for growth, and for reproduction. These rates of change are modified by internal components such as age and size, and external factors such as environmental temperature and food availability, which in our case is light availability. As it models the reserves explicitly, it is ideal to capture the reserve-relying growth mechanisms of Laminaria. In our model, the uptake of carbon and nitrogen are influenced by the environmental irradiance, nitrate availability, and temperature. It is also affected by the size and shape of our individual. It is also important to note that stoichiometry, so that's the balance of the chemical components inside our individual, it's really a key component of our model. So our structure has a fixed composition of carbon to nitrogen, while the reserves are allowed to fluctuate, which is why we model them separately. The model is also capable of defining how much energy that is not used for growth can then be used for reproduction. So how does our model then go from how much light we have to how much our plants grow? So first, energy is taken from the environment. Then it is assimilated into the reserves. Then we have to enforce our stoichiometric balance of the structural compounds. So we have our tissue with the same composition. Before structural growth occurs, though, metabolic cost needs to be paid. This will be higher at higher temperatures, as basically you have a faster metabolism. If the maintenance cost cannot be met, the individual fails to grow and can assume to have starved. The amount of energy not used for growth will then be assigned to reproduction. So you basically have energy going into structure or into maturation, according to depth theory. So once we've got this brilliant model set, what do we have? We've got data that needs to be fed to the model, so we actually have some bases in the real world. We can separate the data that we required in 3 sections: parameters, driving and fitting data, parameters, driving and fitting data. The information that we require for parameterization of the model behavior comes from the allometric relationships and independent estimates for metabolic rates that are from previously published experimental results. The previously mentioned irradiance, temperature, and nitrate availability are our required environmental conditions, which is basically how we understand how the environment affects growth. And this can be used to define where or when our plant simulation is occurring. And that's the main component that we're using to then see the effects of climate change. So after we've got all this data, we need to fit our model. Here we show in green a collation of multiple papers that are documenting the weight of Laminaria hyperborea at different ages. Each of those points is an individual. But each of those points also comes from various depths and various locations, which is what's causing that huge amount of differences in the points that you're seeing at each age. We are also showing the internal composition of carbon and nitrogen at different times of the year for some Norwegian populations, as we have a very seasonal species. So then we move on to what climate change effects are going to be into our environmental data. To do this, I'm here showing you the degree of change that will be expected in each of our environmental conditions on the various climate change predicted scenarios. So these values come from two future scenarios, the RCP 4.5 and the RCP 8.5. We consider them for both the near future, so that's 2040 to 2069, and long term, 2070 to 2099. This is from previous IPCC reports, not from the latest ones. And unfortunately, the previous conditions were a bit more optimistic than the current ones. But very roughly, we see an increase in temperature and irradiance and a decrease in nitrate availability. So we obviously needed to focus somewhat to parameterize our model. So here I'm showing you the different locations of the different studies that we have collated data from on the left. And on the right, I'm showing you the two locations for Scotland that we chose to parameterize the model for. Here are some rough results. Here is some data that we're parameterizing our model to. So this is data from a thesis published in St. Andrews by Jupe in 1972. And due to this wealth of data in this thesis, which is absolutely fantastic, we decided to then focus on Scotland for our parameterization. What you see here is a snapshot of various individuals' weight from different depths and different populations. This is not a growth curve, right? You cannot track that first individual growing until it's 10 years old. What we see here are individuals that were all sampled at the same time point and all weighted at the same time point. Why is this important? Because this means that these individuals have grown under different light conditions. This also means that they're competing among each other for resources. Most importantly here, we can see a clear effect of depth on the overall size of the older plants. As there is a rather large size difference between the young plants and the other ones, we can zoom in to have a better look at the younger ones. So if you look closely, you can just about see that the plants at 3 meters are actually smaller than those at the deeper levels. Our hypothesis for this is that the effect of those significantly larger canopy plants at 3 meters are drastically reducing the light available for those understory individuals. But alas, we do not have 10 years of light data measured in the understory of kelp forest in Arctic. Although if anyone has it, do please share it with me. So there's another amount of data that we found that comes from the Isle of Man. I would like to mention this data from Joanna Kane, as it is also an amazing source of information, as she sampled data from 3 different countries and from various sources in the Isle of Man. So the reason I'm showing you this is just so you can see what the wide range of variations you can see in individual data. So before I was showing you the mean and the maximum and the minimum, here I'm showing you the raw data. So this is the measurements of various individuals. It shows the whole plant weight at different ages. You can see deeper plants shown in blue and shallower ones in orange. There's 3 locations shown roughly in their own column. We have Scotland, Norway, and most of them coming from the Isle of Man. The histogram that you can see above refers to the abundance of the individuals at each age, and you can see that we have more younger individuals than older ones. And again, the high sensitivity of these plants to environmental variations is why you can see this wide distribution of the data. And it is really a key factor that we have to keep in mind when comparing our model to this empirical result. So this is how our model looks. After all this said, here in black, I'm showing you the simulated growth for a single individual of Laminaria hyperborea growing in Scotland at 3 meters depth. The first plot in the top shows the mean weight for each age from the Jupe data that I previously showed you in green and our simulation in black. The bottom plot then shows you the carbon to nitrogen ratio of the plant during 2 years. And it shows you a seasonal cycle that's being repeated on how nitrogen and carbon change. The face plot to the edge shows you the change in carbon to nitrogen content in the plant. The line progresses as time moves on, and the color of the line represents the changing season. The top purple side of the plot represents the conditions when the individual is nitrogen depleted, and the bottom is the opposite. As you can see, the organism internal composition fluctuates on a yearly cycle that stabilizes later on. Looking at the weight, we see that our model recreates the weight of the older plants. This is what our model considers to be the stability point. As I mentioned before, job data is not a curve. It's a snapshot from a single time point, while ours is a curve because we're only tracking a single individual. Basically, our individual has not had to compete with any other plants for resources as the plants sampled in our sake. These ideal growth conditions from our simulation are what we hypothesize is causing the divergence that we see when the plants are younger. This is even more important when you consider the older plants are actually reducing the light for the younger ones. So what happens when we model deeper plants? We move on to simulate a plant growing at 9 meters, and we're comparing it for the empirical data that we have for that depth, also from Jupe's thesis, also for our sake. The key aspect here is that the curve is similar, but the plants growing deeper are smaller than ours. The process behind this is light availability. In our plant, we see that adaptation to the lower light conditions is done by having a lower carbon reserve, which you can see in the bottom plot by having a flatter carbon to nitrogen ratio. Again, our plant is not being shaded at any point, so it's taking all the light that it can get at that depth. The plants from Arazic forest not only have to deal with significantly less light than the ones at 3 meters, but they also have to deal with the older individuals, further reducing it. Again, the phase plot here shows the amount of carbon in the plants. It has just shifted to lower levels compared to the previous ones. Further work right now is being done developing a forest model that will test the effects of shading on our plants. But some preliminary experiments have shown a reasonable response to changing light conditions, which approximate the empirical data that we see. Now we move on to climate change. So how is climate change going to affect Laminaria hyperborea in Scotland? It says these simulations were done for 3 meters. Once we were confident the conditions were being accurately represented. Here I'm showing you the RCP8.5 simulations that are described by the Intergovernmental Panel on Climate Change, so the IPCC, as a moderate scenario in which the emissions peak around 2040 and then decline. Here we're showing you the midterm effects. So this was for 2040, 2069. As we can see, there is an increase in temperature in the table in the left. There is an increase in irradiance, a decrease in nitrate. Laminaria growth under these conditions has shifted to significantly smaller plants, but with a similar seasonal fluctuation patterns in carbon and nitrogen. This limitation to the steady state size of the plant in our simulation is caused by the increase in temperature, as this has a direct effect on the increasing metabolic costs for an individual. So basically our individual is having the same amount of nitrogen, the same amount of light, but it's running hotter. And when it's running hotter, it kind of needs more food. Meaning that in this hotter scenario, that energy that could have been used for growth is being used to just stay alive. And that's why we're seeing a small plant. So when conditions get worse, for here, we're showing the RCP8.5, which is the highest baseline emission scenarios in which emissions continue to rise throughout the 21st century. Again, this is not the latest IPCC projections, but the previous ones. Here, the environmental conditions are even less ideal than in the previous scenario, and as such, we can see the same pattern of a smaller plant that has not reached its expected size. So long-term moderate emission scenarios, we can see the pattern is being repeated. We can see that there's less than ideal conditions, the increase in temperature, we see smaller plants that are really not doing well. but they are still maintaining the integrity of the structure of their components. Worst case scenario, RCP 8.5 for the long term. This is the simulation with the largest temperature increase, which, as I mentioned before, it influences the internal processes of our organism through an increased metabolic cost. This plant has barely managed to assign any energy to growth, but it is capable of maintaining its internal stoichiometry. And we are seeing a plant that could basically be considered to have starved. In conclusion, Laminaria hyperborea growth can be simulated with a process-based model with a sparse number of parameters. And if you like maths and modeling, this is actually really exciting. And this is being done by understanding the balance between internal and external biochemical composition of the plant. This has excellent implications for future incorporation of this model into forest and ecosystem models, as it really facilitates maintaining accuracy and it limits the computational power and the data required to utilize it. Unfortunately, things are not looking good for Laminaria hyperborea in Scotland in a warming world. Our simulation shows that the individual fails to reach even half of their maximum size as they grow to presently, with this response getting worse as the conditions get rougher, understandably. Although the plant is maintaining its internal carbon and nitrogen composition, showing that not even this really resilient complex strategy is enough to mitigate against the worst effects of a warming planet. Thank you very much. Any questions?
Great, thank you so much, Alex. Yeah, as Alex just said, if you have a question, please do type it into the Q&A box at the bottom of the screen. I can see one's already hopped in there. Oh, wait, no, just a great presentation, that comment says. So that's always good. I think that's from a supervisor of yours, actually, Alex. I have a couple of questions myself to kind of kick things off whilst I'm sure our audience are typing questions. So one of them was actually, I can see that you've obviously acknowledged some of the colleagues that you were working with in Cantabria, and I was wondering, did you collect data that you're going to be analysing, like data from that location? Was it just that kind of skill set that you were kind of, what were the colleagues that you collaborated with? What was that work really? Was that just kind of learning what their modelling techniques and stuff like that?
Yeah, so unfortunately, you haven't actually had Laminaria hyperborea growing in Cantabria in the past 50 years because climate change is fun like that. So the skills that I've developed here have mostly been helping in their work. So it's more about learning what's the actual process of collecting data in this sort of diving, dealing with tides, environment, all that sort of things. So for Laminaria hyperborea data collection, it's actually quite difficult because you need scuba diving and they're also quite deep plants. So the funding for collecting the sort of data that this experiment needs is quite large. And if we could have ideal data, we would need about 10 years of data, which is kind of a dream.
Yeah, fair enough. Okay. There was a word in one of your slides that, you know, I haven't got a background in this subject, which hopefully you can explain. It was in your diagram of the process of your modeling, which was stoichiometric.
Yeah,
maybe I might completely fluff that up, but I was wondering if you could just provide for people like myself what that word means, please.
So in a nutshell, stoichiometry just means keeping the chemicals in balance. Okay. It's just a fancy way of putting it.
Okay, that's fair enough. Right, I can see that we've got questions submitted by our audience, so let's hop into that. The first one is from Kelly, and she wonders if you thought if ocean acidification elevated CO2 would reduce the negative impacts of temperature with more CO2 available for photosynthesis. There's a second part to the question which asks, could that parameter be included easily in the model, or does it require a lot more prior lab research?
So, multiple parts to answer that question. Basically, no. So more CO2 won't fix it, because what our model is saying and our model might be mistaken because, all models are wrong, but some models are useful, is that being alive is more expensive when it's hotter. That's all we're saying. So the plant is not really finding carbon to be limited. So having more carbon in the system wouldn't really fix it. Having more nitrogen in the system might have an interesting effect, but as we all know, working in marine science, nitrogen is a limiting nutrient in the sea. But that can be included. It is not included in my model, but it would not be hard to include. Requiring prior lab research, there has been experiments done. So I think you could just use the data that's already published out there. That's basically what we have done for other stuff.
Okay, great. And next is a question from Mike. Does the predicted decline in size with warming match the geographical pattern in plant size in this species? A follow-on question is, and is this the same thing happening now at the warm range edge?
It's another really interesting question. So we haven't actually looked at geographical distributions with the plant currently, because we've sort of more focused on being able to simulate growth given the current environmental conditions. So is it happening in the warm range edge? It is the next step that we have to test in the model. So basically we've got data that I showed you before. So we've got data from Norway, we've got data from Scotland, that's the one I just showed you, and data from the Isle of Man. So I guess a way of answering that question is going to be once we apply the model to these other geographical locations, and we could see that variation in weight from Norway to Scotland to the Isle of Man. And we also have some data from Normandy as well. So that might see if it does work for warming. It seems like it does, but again, rough research needs to be checked.
Okay, great. And Kelly's followed on from their previous question saying they were curious about what other work you've been doing as part of your PhD. Will you or have you done any modelling of other species as well?
This involved a lot more work than it sounds like, just presenting a few equations. So I haven't really focused on anything else apart from trying to understand Laminaria. So this has really involved a lot of reading to sort of learn all of the sections of the life history of the species, how all the sort of environmental factors might affect its growth. There's been a lot of reading and a lot of hunting for data. So as I said, the kind of data that we need to parameterize this model doesn't exist. So we've basically have to play detective to see things that are already out there. Fortunately, there are some brilliant people that have done wonderful research and have their data publicly available, either in papers or the thesis. So we're using that. I won't be using my model for other species, but the brilliancy of the depth modeling is that it should be easily adaptable to other species if anyone wants to go and play with it. Basically, depth modeling means that those few parameters that we chose, that we parameterize with the data that we found, can just be altered, can be tested, can be parameterized, can be fitted to other data. So you could use it for other species, but you need those species to have a similar cycle. So as I said, Laminaria hyperborea is a strongly seasonal species. So it grows in the winter. I am not entirely sure how many species have their fast growing period at the same time or when their reproduction is happening. So there is a lot of rather specific components that might need adapting before you can translate it to other species. But theoretically, yes, it can be done.
Okay, great. Yeah, I think it sounded like a lot of work that you were presenting, even if it was just on a handful of slides. Kelly's followed on from that comment saying, sounds like a lot of work. It's a cool result. I can imagine it has relevance to stable isotope analysis as when you collect samples, which may affect the carbon-nitrogen ratio.
Yep. So depending on what time of the year, the data that you're going to get is different. So the plant's composition in the summer is different to it in winter. And collecting data in winter, as you'll probably know better than me, is not particularly pleasant. So there is a bit of a limiting factor on having data from stabilized analysis from samples collected throughout the year.
Okay, great. We are just coming up to the end of the session. I have one more question. You know, I'm not a Laminaria expert, and obviously you've shown that as temperature, as long-term predictions of temperature increase, the plants are not faring so well. Obviously they store carbon, but are there any other kind of economic impacts that might happen from this plant not doing so well? Is it something that is farmed or is used for other things? I'm just wondering, you know, other than the plants are getting smaller and not doing very well, what else could this mean?
So what will happen is basically what Michael Burris was saying earlier, is that it's going to shift its distribution, shift its range. So it's what's happening in Cantabria, it's what's happening in Spain. You don't have them anymore. So obviously when the plant is gone, because it's too hot for it to survive, it's an environment that you've lost. And they're quite important species at a level of biodiversity. So not only for themselves, but they're also just host a wide range of epiphytes, and they act as fish nurseries. They also have effects on managing wave action. Not my area, but I've read some stuff about that. And also they can be harvested. They're not currently farmed, but they are harvested for pharmaceutical products or raw material, things like alginate. That's used as a thickening agent, quite popularly.
Okay, wow, I've learned a lot of new things today. So that leads me to say thank you very much, Alex, for being today's MASTS webinar presenter. If anyone has a question for Alex that you kind of want to follow up on, her email is actually on the screen there in the top right. So do drop Alex a line if you want. Yeah, she's waving her mouse there, and also her Twitter there. So if you want to give her a follow and see the things that she gets up to, then follow her on Twitter as well. Thank you very much, Alex, for being today's presenter. If anyone wants to follow up on this recording, then please do head over to the MASTS YouTube channel later today, where you'll find the recording there. I just want to highlight as well that in 2 weeks' time, we do have another MASTS webinar presenter webinar happening with Charlotte Lee. If this is also a talk that you like the look of, then please do join us for that, and you can sign up. So thank you very much, Alex, again, and we hope that you can join us for a future webinar.